What I'd like to do today is to cover first a little bit about the companies, just to orient you, then to talk about how we're enabling clinical centers in order to manufacture their own CAR-T cells, then move on to focus on lentiviral vector manufacturing and our manufacturing methods and how we've moved away from an adherent process to a chemically defined serum-free suspension manufacturing process. And finally, something that we're doing, a little bit of work that we're doing on creating reference standards. So Miltenyi Biotech, we're a 2,500 person company, global operations, headquartered in Cologne in Germany, 500 scientists, 14,000 products, starting with magnetic cell isolation, but more recently devices, automated devices, and then also vectors. And we were acquired Lentigen with the idea of integrating our competency, lentiviral vector manufacturing, with the automated cell processing that Miltenyi already had resident. And we're located in the suburbs of Washington, D.C. We specialize in lentiviral vector design. That's a very important feature. How you design the vectors really depends upon titer and performance. Then we also make preclinical vector and GMP, all the way from clinical to commercial. We have currently a 34,000 square foot facility. We're expanding. We just purchased a 150,000 square foot facility across the road. And our most recent success has been to move from an adherent to a chemically defined serum-free suspension manufacturing process that leads to very high titer vector production. On a good day, previously in an adherent culture, you get 1 by 3 by 10 to the 8. Now we get 5 by 10 to the 8 to even 10 to the 10 transducing units per mil. This results in hundreds to thousands of doses per lot, depending upon the vector, and of course a dramatic reduction in cost per dose. So how do we work with investigators? Two types of investigators. Those are companies. It's a fee-for-service. They have their payload gene. We put it into our vector. We contract manufacture for them. With clinical centers, we have our own portfolio of lentiviral vector CARs that we provide to the clinical sites without cost, so that they can run their own clinical trials. They have to use the Prodigy and the entire workflow that I'll just describe to you in just a moment, but it's enabling clinical sites so they can manufacture and innovate by combining what they know with the workflow that we have developed. So this is the current way that CAR-T cell products are produced. You start with a leukapheresis product, you select your cells, PBMCs or a subset of those cells, you stimulate the cells. Generally today it's with the Dynel hard bead stimulation reagents. Then you transduce the cells either with a lenti or a retroviral vector or another vectoring system. There are a couple out there that are in use. And then you expand the cells in medium and a cytokine combination that you use, usually IL-2. You can see down there the devices that are used for the unit operation, the highly technical trained personnel that are required to generate these products, and then the GMP facilities in which each room has to be dedicated in order to manufacture each patient-specific product. Our vision is a little bit different, that a device does the heavy lifting. We also start with a leukapheresis product. It's loaded onto the device. The device is a completely enclosed system. That means the product doesn't touch the device. There's a plastic manifold that comes on top of the system, which generates the cells. The cells are grown and selected for completely within the system with little operator intervention. So in our case, we isolate 4s and 8s, and we think that's a superior product. I won't have time to go into why that is in this talk, but it's a superior product compared to other products. We don't use the hard beads. We use the Transact. It's a soft dextran ion matrix that contains mobilized CD3 and CD28. We have shown that you enrich for a T central memory phenotype when you use these beads or these matrices. We use exclusively a lentiviral vector, which I'll show you in just a moment, and then we support the T central memory phenotype with an IL-7, IL-15 medium. The Tex-Mex medium that we use is completely now serum-free. We had to use a little bit of serum until a couple of years ago. We're able to identify the factors that are now needed, and it's a completely serum-free closed system process. This is just to show you some data here, whether it's a healthy donor or patient material in yellow. You can see the starting material is in the 10 to the 8 cell range. And you can see on the green, the far right, it's a very robust process that you generate almost always approximately 10 to the 9 CAR-T cells for your final dose. And that's the sort of dose that you require. So this is the current state of the art, the manual manufacturing method. You require many highly experienced staff. And as you can see, as the numbers of doses increase, it gets very, very cumbersome to handle this. And that's why they're done in centralized facilities. It's very difficult to conceive of a manual method in a decentralized mode. So that's why they're in large factories. But they're difficult to scale for large numbers of patients, and they're almost impossible to scale beyond a single site because of the issue of comparability. Our vision is, again, different. Instead of having 2 to 3 operators per product to generate a single product, one operator can be monitoring 4 to 5 machines, 4 to 5 machines simultaneously, which significantly decreases the labor cost and the overall product failure rate, and it provides options to manufacture these products beyond a central manufacturing facility. So on the left there, you'll see the centralized mode, the hub mode, in which you take cells and you ship cells to a centralized plant. The products are made and then shipped back. Automation, the robustness of the automation allows you to start to think about making the cells either on the top right within the hospital or close to the hospital. And it will differ from region to region. Sometimes they'll be made at the hospital, or one hospital may, or a couple of hospitals may share a manufacturing facility, like in some places like in the Middle East, where I flew from last night. So the FDA and the EMA, we're in close discussions with them, and they are very supportive of this approach, but show us the data, and they're quite right in that. And in addition to IIT trials that we have initiated globally to collect the data, we are running a definite clinical trial in Germany in order to show product comparability. So this is a German trial using our 19 CAR vector to modify CAR-T cells, and they're going to be manufactured in 12 sites in Germany, and they'll feed from in 7 sites in Germany, and they will feed from 12 clinical sites around the country in order to from which the main goal is to investigate product comparability and to accrue outcome data. And what's useful about the Prodigy is that we're dosing fresh in and fresh out. That means the cells come in fresh to the site, they are manufactured, and they go back out to the site fresh on ice. So it's a little bit different than the current manufactured products. You have to always have an option for freezing, just because the patient may, say, come down with an infection during the manufacturing process. So you have to have the option to freeze, but generally it's fresh in and fresh out. And so right there in purple on the bottom, we are creating a global IIT network, and now we're in each continent or each region initiating clinical trials to, again, find supporting data for this decentralized methodology of manufacturing CAR-T cells. We exclusively use lentiviral vectors because it's a clinically proven workhorse. They're very efficient. You can get very high levels of transduction. You've got an excellent gene payload capacity. You don't need excessive stimulation. In contrast to gamma retroviral vectors, they're very stealth. Numerous studies have shown that there is no genotoxicity associated with the backbone of the vector. And that lack of genotoxicity is also observed in the natural infection of HIV. Over 30 million people infected with the virus, billions of integration events, and yet not a single evidence of a T-cell leukemia due to genotoxicity. I think that's very good world evidence to show the safety of these vectors. So we've moved away from the adherent serum method of manufacturing. That's on the top left. They used to use these cell stacks and use serum medium, and now into a bioreactor format, just like monoclonal antibodies are made. You start with a train, you can see there in the center. You scale up into these large bioreactors, anywhere between 50 to 200 liter. That's our current mode. And these generate very good titers in the, as I mentioned, 10 to the 8 to 10 to the 10 transducing units per mil. This is just to show you a little bit about the titers that you get from different lots of vectors. Each color is a different type of vector, and you can see that within each color you get reproducibility of the lot production, but you can see that the titers and performance of these vectors can differ quite substantially. And so it's very important when you design a vector to really consider what is your target product profile in order that you sort of match that with what you're trying to achieve. But sometimes a high titer vector or a high expressing vector is not necessarily what you want. Let me give you an example. We had a client that developed a CAR, and we cloned it into a couple of different vectors. And out of the several vectors that we created for them, there were 2 that were sort of honed in as the ones that they really wanted. One expressed the CAR at higher levels, the other one expressed the CAR at lower levels. You'd think that the client would have selected the vector with the higher level expressing CAR. No, they chose the other one because the higher expression of the CAR resulted in tonic signaling of their CAR molecule. And they didn't see that tonic signaling with the lower expressing current, so that's what they chose to move forward into the clinic. So it really depends. The titer is not the key thing. It's your target product profile that is key. So we have two formats for manufacturing GMP vector, 50 liters and 200 liters, but we've also developed a scaled down version of the process at 4 liters. And that's very important because you don't want to make the investment in 50 liters or 200 liters until you've done a scaled down version to show that your vector is working, because it's less expensive. So you can see there on the data on the right, whether it's a harvest supernatant or the final fill, you can see the titers there in the multiple 10 to the 7 range, and then in the final fill, you can see anywhere from the 10 to the 8 to the high 10 to the 9 range. And actually you get we were expecting that it would either be level or maybe it would be a decline, but actually we're seeing even a bump in titer as you scale up. And it makes a lot of sense that when you take serum out of the process, it makes chromatography a lot easier, right? Than have serum to clog up the chromatography process. So this is how we test the bulk plasmid DNA. You can see, I'm not going to go through the list there, but you can see the 260-280 ratio. The identity is very, very key. Sometimes when we get a plasmid from a client, it doesn't always match to the actual identity of what they're hoping that their product will be, so that's very, very key. Obviously, sterility is very key as well. For the GMP vector, the testing, again, you have your various sterilities. Now, from a guidance point of view, we have to test for RCL at 2 stages. So that allows the clinical sites to only use a PCR assay to assay for RCL. So at the clinical sites, they only need to assay for residual VSVG. But that's why the manufacturer of the lentiviral vector has to test the RCL at 2 points, at the harvest and the end of production cells. Genomic integrity, that's very important. You transduce cells, and then you show that your gene has integrity by sequencing. Residual benzenase, and you can see the appearance and fill volume. They're usually also very important parameters. So moving on to our candidates that we are moving into the clinic, that we have in the clinic. This is a CAR-19 construct that we have developed. It's the FMC63 binder. It has the 4-1BB or CD137 co-stimulatory domain. And then we have a slightly different transmembrane domain from the TNF superfamily. The various constructs there, the 1494 construct is the construct that we made for Carl June for that New England Journal of Medicine paper. The 1563 is our construct with the that's configured right there. You can see in the Raji cells all the various constructs work quite nicely, and on the 293 T cells they don't kill as a control. The animal data here, you can see that the 1563, when the animals are challenged, so these animals are first injected with a tumor and they light up, by analysis, and the 1563, when you challenge the cells, clears the tumor. The 1464 also does a very, very good job as well. So we're now into the clinic in 2 sites in the US for adult B-cell lymphoma. This is a clinical trial being led by Marcus de Lima, who's the central person there. The thing I want you to notice is that the prodigy, the enclosed system, is not in the clean room. The clean room is behind them. This is the outside area. That's still an ISO 7 regulated laboratory, but it's not a true clean room. Right? That's how they manufacture the product, and they're using normal lab coats. Because it's an enclosed system, right, there's a lot of benefits in terms of ultimately the cost to manufacture. You don't necessarily have to run a full clean room. So the clinical trial to date, the results, we've got 4 out of 5 patients in complete remission, 1 progressive disease. Almost in all cases, and this is, I think, important, we're not seeing either CRS or neurotoxicity of greater than grade 2. And we think part of the reason for that is that the TransAct nanomatrix that we use to stimulate the cells generates a lot of T central memory cells rather than a lot of effector cells. That may be part of the story why we're not seeing toxicities of greater than grade 2. And as I mentioned, this is done in a normal laboratory that is ISO 7 qualified. We also have a first-in-human pediatric clinical trial for ALL being run at the Dmitry Rogachev National Research Center of Pediatric Hematology, Oncology, and Immunology in Moscow, in the Russian Federation. And out of 21 patients dosed, 19 are in complete remission. And again, no toxicities of greater than grade 2. So we were interested in the whole idea of a bispecific CAR, right? And we are focused on developing a tandem 19-20 construct, if the idea is 2 better than 1. And you can see there the types of constructs that we had developed. You have on the top there 2 19 constructs, then a 20 CAR. They all contain the 4-1BB. Then a 2019 CAR, and then a 1920 CAR. And on the bottom right, you can see a depiction of the 2019 CAR that we eventually took into the clinic. So on the left there, all the constructs work very nicely in terms of killing Raji cells or NALM6 cells. You can see the killing, the specific lysis there on the top. In the center are the control cells. No killing, 293 cells or K562 cells. We always like to include K562 cells because they're a noisy cell line. And if we see 2 constructs and one shows a high specific activity on K562 cells, we will usually select the other one. It's a useful cell line. And then we created 2 different cell lines from the K562 in order to show the specificity of each construct, because the FDA wanted to know that the constructs that kill 19 can kill 19, and those that have 20 can kill 20. And these are OR constructs. That means only one of the 2 antigens are required in order to get killing. So on the bottom left there, K56219, you can see that only the constructs that expressed 19 killed. You can see the orange, the 20 did not kill on the bottom there. The constructs on the right there, the constructs that expressed 20, they killed, and the 2 constructs, the 19A and 19B, did not kill, showing the specificity of these constructs to basically kill specifically their target cells. This is the animal data. Let's just focus on the bottom left there, which is the day 32 data. I don't think you can see that. So you can see that the 19B construct cleared the tumor very well. We have the 2019 that also clears, and the 1920 also clears. What's interesting is we have this 19A and 19B constructs. The only difference between the 2 constructs is just a linker, right? But you get a very different performance. Even by a simple linker can affect the performance of the CAR. But this doesn't answer the question of whether a 2 is better than 1. And so our team members, led by Dina Schneider, developed a high tumor burden model in which to understand that if we can start to see breakthrough with a 19 construct, will that be ameliorated with a 2019 construct? And you can see on the panel B, the 19B, you're starting to see breakthrough in the center there, while the 2019 you don't see the breakthrough. So at least we have a hint or a handle that perhaps 2 is better than 1 in an animal model system. But nevertheless, we have moved over, and this construct is now into the clinic at the Medical College of Wisconsin. Again, I'll focus your attention on the right there, that the prodigy is in a laboratory. It's an ISO 7 qualified lab, but it's not in a full clean room. They have dosed now 13 patients. Overall, 8 out of 13 patients have seen a complete response. If we go to point number 2, the response by dose level, you'll see at the lowest dose, we had 1 complete response out of 3. At the second dose, 1 complete response out of 3. And then at the third dose, 2.5 by 10 to the 6 cells per kilogram, 6 complete responses and 1 partial response. So very, very excited about that. And in fact, our collaborators at the Medical College of Wisconsin were quite excited that they put the first patient on the news there. You can see a little cutout there. And the patient there, Brett Carroll, it's a very interesting case. A 52-year-old with mantle cell lymphoma, had stable disease coming into the clinical trial, but as his cells were made, he deteriorated very, very rapidly. This first patient, they were going to freeze the cells and then dose. But it was very, very clear that this patient wasn't going to survive if they were going to freeze the cells before dosing him. So the investigators got a dispensation from the FDA to dose the patient fresh. They did that, and after they dosed the patient fresh, Brett, after 1 week, he wanted to go home, and 17 months later, he's in complete remission with minimal residual disease, no detection of MRD. So we're very excited about that. That's actually patient number 1. So this is a little bit of more details about the patients in the clinical trial. So you can see the various diseases. You can see that in prior lines of treatment, heavily pretreated, lots of different lines of treatment. And if you look at the fresh versus frozen, until we changed the protocol, there was some fresh and frozen, but now we're completely dosing in and fresh out. And what's interesting so far, although it's very early, maybe it's coincidental, but so far we've seen that all patients that have achieved a complete response at day 28 remain in complete remission to this date. So we're excited by that, but it's early days yet. And this is just the data to show the superiority of fresh product over frozen product. Here it's a 20 CAR-T cells. And you can see while frozen and fresh work, by day 20 they're all working fine. You can see there's a lag of the frozen cells in order to reach their full benefit compared to fresh, which have their effect immediately. And that window may be important for certain patients. So to release such a product, a decentralized product, there are a couple of things you have to take into consideration. You have to, each site, in a decentralized model, each site will use their own local sterility, whatever that is, whatever they're using for BMT, to release the product. Then there's a duplex PCR assay for copy number and VSVG that we provide to the site without cost. And then there is a fax analysis profile. We're recommending sites to use the MaxQuant because the data can then be harmonized globally. And then there are express modes that are self-gating and self-calibrating in order to generate reliable data that can be compared from site to site, because the data, the self-calibrating and self-gating mode allows for less variability of the data. We're establishing a global network of these manufacturing facilities, and that's their facilities, they're not our facilities. Each site owns everything themselves. They only need to go into a clinical trial with us, only with our vectors, but they're free to collaborate with anybody, with any vector to make CAR-T cells locally. And we took a page out of Apple. The Apple had their user group meetings. We started our own Prodigy user group meetings, in which enables clinical investigators to meet and discuss and exchange ideas on how to create this network. Finally, my last section is some work that we've done to develop some reference standard cell lines, copy number reference cell stand lines with lentiviral vector copies of either 1, 2, 3, or 4 copies. We've been able to generate data to measure the copy number either by next generation sequencing, by a GAG duplex qPCR, or by a C-fragment PCR. You see the data is very, very consistent there. We've also been able to map the integration sites for the various clones and show that the stability of the standards of the genomes within the cells over a longer period of time. So we are actually collaborating with USP and EDQM to eventually make these the DNA available for all as a reference standard. I'm a little bit ahead of schedule, I see, but I just would like to acknowledge the patients that are on our clinical trials, the collaborators there, Terry Fry, Crystal Makel from the NCI, Niraj Shah and Hari Parshwan from Case Western University, Marcos de Lima and his team, and Michael Marchand from Moscow. Also, special thanks for the Miltenyi Biotech team. The German team did all the hard work in terms of making the Prodigy very, very robust and enabling this ability to use these devices in a decentralized mode. And then my team there on the right, who are a superb team. Thanks very much.
Thank you very much for this great presentation. It's open for questions. I guess I'll start. So in terms of the criteria for consistency between sites, as we know in the autologous world, every patient is different and every manufactured product has different characteristics within the specs of the QC. So what is exactly the criteria for consistency that you can put forward to demonstrate to the regulatory agencies that there is a consistent manufacturing product?
Yeah, so I didn't go into it in great detail, but obviously reaching your target cell number, right? Your product profile in terms of reaching the target cell number, your actual product profile, the level of CD4s and CD8s, the surface expression of CARs, that's the sort of the major criteria.
Thank you, Boro. Since you have experience producing many different types of cassettes with the Lenti, do you have, in terms of downstream processing for all these vectors, there is huge difference when you change the cassette really for the CAR-T? In other words, do you have a platform that you can really plug and play, and particularly at GSP level, or you have to really adapt a lot from vector to vector?
Yeah, no, it's a single platform for manufacturing. We don't change our downstream process. We would rather put into our family of vectors the construct and then they produce their various titer and the target product profile of the vector. We don't change our manufacturing process. It's a transient system. It's not a packaging line, but it's a set system. Yeah. But we do find that when you change certain elements, promoters, configurations, you can vary the titer greatly and the performance characteristics. And it's at that level we focus on trying to optimize the constructs rather than the other way around.
And DSP particularly, so it behaves similarly?
Similarly, yeah.
Great.
Hey.
Hi, Boro. Congratulations, nice presentation. Lots of great progress. So I'm curious about the Prodigy, so the containment level. Really, the unit contains material, it's professional grade material, but it can be operated outside of a highly classified environment. Is that correct? Or just to understand, does that reach forward to a BLA level, would you anticipate?
So it's at the clinical trial level at the moment, right?
Yep.
And we're generating data.
Yes.
And we haven't reached the commercial level yet.
Right.
And we'll see.
Okay. So in discussion, in consideration.
In discussions, exactly. But
Thanks.
yeah.
Yep.
Yeah, please, if you can use the microphone.
Can you please comment on the reference standard that you have generated with JARCUT, if it can be used for titering, because we use 293 T cells for titering, and we know that it's a polyploid cell, so if it's comparable?
Yeah, it has a normal, it is very close to a normal karyotype, so that's why we picked the JARCUT, and that's exactly, it's to create for your validation of your PCR protocols, so that you can get a reliable result. That's why we created it. Yeah.
Yeah, one more question. So in terms of the fresh versus frozen products, obviously you see a different slope in response, although it seems to normalize after a few days. In terms of the length of the response in your animal models, do you see a difference eventually past day 20? And what does that tell us in terms of dosing the patients? Do we need to do some adjustment in the cell dose based on the characteristics of the products tested in animal models?
Well, clearly there's a window of opportunity there for fresh product over frozen. And that allows for us, the investigator or the clinician, to have options, right? So if the patient really needs the cells immediately, they can dose the cells immediately. Or if the physician wants to create 2 or 3 doses, they can create 2 or 3 doses. So it allows that flexibility. We didn't see any difference after the effect. Once they cleared the tumor, that was that.
Okay, thank you very much.
Thanks.